After watching this video, you should be able to describe the hormonal regulation of glycolysis, particularly in the liver. Now let's start with a big picture and understand that when we're talking about hormonal regulation of glycolysis, or really any major metabolic biochemistry pathway, and we're considering fasting and the well-fed state, we're really thinking about the effects of insulin and glucagon. Glucagon being high in the fasting state and insulin being high in the well-fed state. Now, if you remember, there are three irreversible steps of glycolysis. Steps 1, glucokinase, which takes glucose to G6P. PFK1, which takes fructose 6-phosphate and makes fructose 1,6-bisphosphate, and pyruvate kinase, which takes phosphoenol pyruvate or PEP and makes pyruvate. And it's these three irreversible steps, steps 1, 3, and 10, that are the ones that are going to be regulated through effects of insulin and glucagon. And when we're thinking about insulin and glucagon and what kind of mechanisms they're going to be using, the two ones that you want to think about are changes in gene transcription and changes in phosphorylation status. So let me give you a couple of examples. When you're thinking about the glucagon receptor, remember the glucagon receptor's GS, it's a G protein coupled receptor, increases cyclic AMP, increases pKa. Now pKa can do some interesting things. It can phosphorylate existing proteins, in this case enzymes, at serine and threonine residues and change the activity of the enzyme. Also, pKa, when it gets activated by cyclic AMP, remember those regulatory subunits that come off that now unmask the nuclear localization signal, can allow pKa to go into the nucleus, phosphorylate cyclic AMP response element binding protein, or CREB, which then can affect gene transcription of enzymes. Now, depending on the enzyme, this mechanism may or may not be happening. Okay, but the CREB would be sitting on a particular gene that pK could be regulating. Okay, when you're thinking about insulin, remember insulin, even though it's the opposite hormone to glucagon, it doesn't use a G protein. It uses a different superfamily of receptors. It's under the category of RTK, receptor tyrosine kinase. And in that case, through a series of complicated steps, you end up with this PI3 kinase pathway, which can affect phosphorylation status as well as have some genomic effects. Okay, so those are the two hormones. They will be using different kinds of receptors, but you can get the same kind of effects. Same general idea. Okay, so now what I like to do is review some things before we go into the details of how these hormones affect these three irreversible steps. And I think what we want to do is we want to start with a schematic that we saw in a previous video that grouped these enzymes together. So we see here on the left, we got glycolysis, glycogen synthesis or glycogenesis, fatty acid biosynthesis, triglyceride synthesis, pentose phosphate shunt. All of these guys are together because when one is on, all of them are on, or when one of them's off, all of them are off. Okay, they all go in the same direction. And you see that they're lined up with the opposing reactions. Okay, so here we're oxidizing glucose, taking glucose to 2-pyruvate. That's happening in glycolysis. Gluconeogenesis takes 2-pyruvate and makes 1 glucose. Okay, so they're really opposite types of pathways. When we're making glycogen and glycogenesis over here, we have the opposite pathway, which is the degradation of glycogen, called glycogenolysis. All right, and we have the same idea for the fatty acid metabolism. We have fatty acid biosynthesis over here. We have fatty acid oxidation or beta oxidation over here, where we're oxidizing the fatty acids. We also have their accompanying associated pathways. They're not really opposites of each other, but we have triglyceride synthesis, which is directly related to fatty acid synthesis because you need acyl groups to make triglycerides, and then you have the fatty acid oxidation along with the ketogenesis because when fatty acid oxidation makes acetyl-CoA, remember those acetyl-CoA molecules are where are needed to make ketone bodies. Okay? And then we have pentose phosphate shunt, which is really off by itself in this group over here. There really isn't an opposite mechanism, opposite pathway on the flip side. Remember, pentose phosphate shunt was important in making NADPH, so you can make fatty acids and also cholesterol. Okay? Now that we have this big picture of the enzyme reactions, sort of grouping them this way, let's take a look at schematically how this might appear in the liver. Okay, and so we have a diagram that looks like this. Again, this was shown in another video where we have the liver cell, and we're showing glucose going in and out of the liver depending on if you're fasting or in a well-fed state. And this is a really good way to think about it. You know, when you're fasting, the liver's job is hepatic glucose production, right? You want glucose to go out of the cell, down its concentration gradient, through this constitutively expressed GLUT2 transporter. Okay, and how are we going to make this glucose go out of the cell? Well, we need to make it, and that's where gluconeogenesis comes in. That's where glycogenolysis comes in. We have fatty acids that are coming from the adipose cells that are going to be oxidized. You're going to make some ATP. You're also going to have this acetyl-CoA, which can go into the TCA cycle, ultimately make electron carriers that can go into the electron transport chain and make more ATP, and all that ATP is really important because we need it to drive gluconeogenesis, and we also are making ketone bodies from acetyl-CoA which go off into the blood and go places like the brain, for example. So this is what would be happening in the fasting state. All those reactions that we talked about that would be turned on would all be up, right? Now, if we think about the other side of the coin, the well-fed state, right, we have lots of glucose in the blood, and we want that glucose to be taken up by cells. In this case, it's going to be taken up by the liver through, again, this constitutively present GLUT2 transporter. And this glucose needs to get into the cell through its concentration gradient. This glucose needs to be maintained at a low concentration, and it's going to be immediately phosphorylated by glucokinase and then finish off its glycolytic pathway. You can go into glycogenesis, pentose phosphate shunt to again make that NADPH so you can make fatty acids and fatty acid synthesis, also some cholesterol, which you would be getting the building blocks from the acetyl-CoA from the pyruvate, and then ultimately make some triglycerides, package it all together, send it out as VLDL so that can be sent off to your fat cells so they can store the acetyl-CoA ultimately as triglycerides in adipose for future use. So this is a schematic then of fasting and well-fed state side by side, grouping all those reactions that like we did in that previous schematic I showed you. Okay. Now, since we're talking about glycolysis, and that would be happening over here, let's take a look at that again and review some of that to help us talk about the regulation. Okay, and so that would be something that looks like this. So now we're focusing on glucose going into the cell. We're in the well-fed state, and here is our 10 steps of glycolysis, right? Starting with the glucose molecule, ultimately leading to 2 pyruvate molecules. Now what we want to focus on are the enzyme reactions, the enzyme steps highlighted in red here. So here's step 1, glucokinase takes glucose to G6P, PFK1, fructose 6-phosphate to F1,6-bisphosphate, and the final step, step 10, pyruvate kinase, which takes PEP and makes pyruvate, right? But those are going to be the major enzymes that are going to be regulated, and it makes sense because these are the irreversible steps, okay? All of the other steps of glycolysis are reversible, and that means that when you're going in the opposite direction, in gluconeogenesis, those enzymes that are reversible, they're going to be used again. And it's really only these irreversible ones that you'll need additional enzymes for that we'll talk about when we discuss gluconeogenesis. One of them, however, we will talk a little bit, that'll be a bit of a preview, is that when you're going from F1,6 bisphosphate back to F6 phosphate, there is a bisphosphatase 1 that will be important. There's an interlinking of regulation with those two enzymes. So that's one enzyme that's a gluconeogenesis enzyme that will preview for you for gluconeogenesis. Now, if we go in and summarize the regulation for these three enzymes, all right, it's going to look something like this, okay? And so we have step one, here's our glucokinase, and in this case, the major mechanism would be insulin induction of glucokinase. All right, remember insulin uses an RTK, it can have effects on gene transcription. In this case, this particular form of hexokinase, remember we talked about in the previous video that there's hexokinase and glucokinase is unique because it's an inducible enzyme, and insulin is the hormone that induces it through the PI3 kinase pathway. And so when we're fasting and there's low insulin, because we don't have any glucose-stimulating beta cells to secrete insulin, glucokinase is not around. And that's good because we want glucose to go out of the liver cell. We don't want it to be phosphorylated, so that's a good thing. When we're in the well-fed state and we have that glucose-stimulating beta cells to make the insulin to circulate around the blood and ultimately activate receptors on the liver, we want glucokinase turned on because we want to facilitate glucose uptake by the liver and glucose 6-phosphate going off and doing all those things we talked about, glycogen synthesis, pentose phosphate shunt, and ultimately finishing off glycolysis. So these effects make sense in terms of the direction. It's just that the mechanism is mostly through this insulin effect on transcription of the glucokinase enzyme. Okay? So that's a pretty straightforward mechanism. Now step 3, which is PFK1, the third step, which takes fructose 6-phosphate and makes fructose 1,6-bisphosphate. This is a little more complicated and involves a new molecule that we haven't talked about, F2,6-bisphosphate. Now this molecule is not an intermediate in any major metabolic reactions. It's simply an allosteric regulator of PFK1, and as we'll see, it also is an allosteric regulator of the opposite enzyme to PFK1, the gluconeogenic counterpart, fructose bisphosphatase 1. Okay, so how is it that the hormones affect this molecule here? And it turns out that it really has to do with pKa. So when you're fasting and you have lots of glucagon around and you have lots of pKa around, the mechanism is that you have a less amount of this stuff floating around inside the liver cell. And as a result, PFK1 is turned off. When you're in the well-fed state and you have suppression of glucagon, remember the beta cells secrete insulin, which can suppress glucagon directly. Also maybe some GABA comes out. You have less glucagon, less PK, and so you have more of this F2,6-bisphosphate allosteric regulator, which then turns on PFK1. Now the details of how pKa affects the levels of this molecule, we're going to need to show another diagram for. But the take-home message, though, is that you're changing the levels of this molecule to change the activity of PFK1. So it's a little bit indirect. Okay? The final step, step 10, PK, pyruvate kinase, is fairly straightforward. It has to do with pKa directly hitting the enzyme. So again, when you're fasting, you got lots of glucagon, you got lots of pKa. PK, pyruvate kinase, is phosphorylated at serine-threonine residues, and the activity of the enzyme goes down, turning off the conversion of PEP to pyruvate. And that's important because we want, when we have glucagon around and we're fasting, we want to be running gluconeogenesis. So we don't want PEP going to pyruvate. Remember, we want pyruvate going back towards glucose. So this really makes sense. And then when we're in the well-fed state and we have low glucagon levels and low pKa, we want this enzyme turned on. So it's not going to be phosphorylated as much, and so you're going to have more pyruvate kinase activity. Okay, so we have examples here of transcriptional effects, changes in phosphorylation status, and this step 3 here, this one in the middle, involves also phosphorylation status, but it's a little bit more involved. It involves PK phosphorylating a new enzyme that I have to introduce to you. Okay, so that's what we're going to do next, is we're going to zoom in and focus on how PFK1 is regulated by pKa. Okay, so that's what we're going to look at. So that would be looking something like this. Okay, so let's take our attention to the cellular picture first, and then we'll summarize things above. So here's glucose going to G6P, that's glucokinase. Okay, that's step 1 of glycolysis. And then we have the second step of glycolysis, is reversible step where we make F6 phosphate. And then here's an old familiar friend, here's PFK1, where we make our F16 bisphosphate. Now I've included on here, which is now a new thing, it's a preview for gluconeogenesis, I've included FBPase 1, okay, fructose bisphosphatase 1, as the opposite to PFK1. So you see all fructose bisphosphatase 1 is doing is just removing that phosphate and helping us go backwards. Okay, and so I added that here because, like I said earlier, fructose 2,6 bisphosphate regulates both of these enzymes together, so we might as well have them together. Okay, now fructose 6-phosphate, in addition to being phosphorylated at the 1 position, can be phosphorylated at the 2 position. And when that happens, that's a little side reaction. It has nothing to do with glycolysis directly. Okay, the fructose 2,6 bisphosphate is not an intermediate in glycolysis. It's just an allosteric regulator of glycolysis and is also gluconeogenesis. And the enzyme that does it shouldn't be a surprise where we get this name from. Phosphofructokinase 2. So it's different than PFK1 because we have this 2 here, and it's because it's making the 2,6 bisphosphate molecule rather than the 1,6. Okay? Now, there's a little bit of a curveball because the opposite to PFK2 is fructose bisphosphatase 2. Right? It's just going to remove the phosphate and make our F6 phosphate again. But the strange thing is that these enzyme activities are part of a single polypeptide. Right? So it's really a bifunctional enzyme, right? And then when one of these enzyme activities is on, the other one's off, and vice versa. So it just, instead of you having a situation over here with PFK1 and fructose bisphosphatase 1, which are separate enzymes, these enzymes are kind of just fused together, if you want to think of it that way. Now, the way this enzyme is regulated is through changing phosphorylation status, and the main regulator of this is pKa, our glucagon signal. So here's our pKa, which we would expect to be around if we had lots of glucagon around, or fasting. And it phosphorylates this bifunctional enzyme, and it turns off the PFK2, and it turns on the fructose bisphosphatase 2. And what that does is it removes this F26 bisphosphate from the intracellular compartment, converting it back to F6 phosphate, and that's helpful for 2 reasons. We're now getting some extra carbon. You know, we can now use that to make more glucose, right? We can remember be going ultimately back to G6 phosphate and then back to glucose and then out the cell. And we're getting rid of this allosteric regulator of PFK1, right? You know, we now have less of F26. We now can turn off PFK1, and we don't want this enzyme on when we're fasting. We have lots of pKa, right? We want to go backwards. We want to go F16 to F6, and that's going to be occurring because F26 also is an inhibitor of F-bisphosphatase 1. So you kind of get 2 benefits, right? By getting rid of this allosteric regulator, we now turn off PFK1 and at the same time turn on fructose bisphosphatase 1, which is a gluconeogenic enzyme. Okay? So now let's summarize this up here. So when we're fasting, we got lots of glucagon around, we got lots of pKa, and pKa phosphorylates this bifunctional enzyme. And what happens is we have less of the kinase 2, more of the bisphosphatase 2, and the combination of those 2 activities means we're going to be making less 2,6 and breaking it down more, and so the levels go down, and we turn off glycolysis and turn on gluconeogenesis. Right? PFK1 goes down, fructose bisphosphatase 1 goes up. And that is going to help everything flow back towards glucose so it goes out of the cell and goes into the plasma. When we're in the well-fed state and we have lots of insulin around and low glucagon, we don't have a lot of cyclic AMP and PK inside the cell. Consequently, this bifunctional enzyme is not phosphorylated, and as a result, the PFK2 activity goes up, the bisphosphatase 2 activity goes down, and now we're making more of this stuff. We're not breaking it down as much, And because it's an allosteric regulator of these two enzyme activities, we're going to get PFK1 turned on and bisphosphatase 1 turned off. Okay? So glycolysis on, gluconeogenesis off. And that is really how this would work. So pKa is really affecting an enzyme that is not part of gluconeogenesis and glycolysis, and it's sort of indirectly affecting that reaction. Okay? So we go back to where we started. Okay, go back over here. Remember, our focus is always going to be on fasting and well-fed with insulin and glucagon, these 3 irreversible steps. Remember, glucokinase was pretty simple. It was affected by insulin levels and induction of the enzyme. Pyruvate kinase was very straightforward. It was pKa, phosphorylation of the enzyme, affecting its activity. And then we finished up with PFK1, which was step 3, which involves that fructose 2,6-bisphosphate molecule. And remember, to put it in perspective, we're always going to be dealing with changes of transcription, like we had with glucokinase, or phosphorylation status, either the enzyme directly, pyruvate kinase, or indirectly, remember, which was affecting that allosteric regulator of PFK1. And that concludes this lecture on hormonal regulation of glycolysis.